In the periodontium, the periodontal ligament is a narrow band of fibrous tissue between two mineralized tissues, the alveolar bone and cementum. It has a remarkable capacity of maintaining its width over time and also to increase or decrease its width according to requirement. This homeostasis of periodontal ligament is maintained by a large group of cells. These cells of the periodontal ligament can be grouped into various categories. All cells which synthesize tissues are grouped as synthetic cells. All cells which are concerned with resorption of tissues are grouped under resorptive cells. Rests of Malassez are the only epithelial cells. Mast cells and macrophages are grouped as defense cells, and the undifferentiated mesenchymal cells are the progenitor cells. We will start with the synthetic cells. All these cells have large number of rough endoplasmic reticulum and Golgi apparatus for protein synthesis and mitochondria for high energy. Osteoblasts. Osteoblasts are the bone forming cells. In the periodontium, osteoblasts are present on the inner surface of the alveolar bone proper. They are cuboidal in shape with nucleus away from the bone. They are in contact with one another by desmosomes and tight junctions. Cementoblasts. Cementoblasts are the cementum forming cells. In the periodontium, cementoblasts are present on the surface of cementum. They are cuboidal to spindle shaped cells. Fibroblasts. Fibroblasts are cells which synthesize collagen for periodontal ligament. In the periodontal ligament, they are uniformly distributed and are the predominant cells. They are in contact with each other through cytoplasmic extensions. They are mostly fusiform in shape with their long axis parallel to the principal fibers of periodontal ligament. Fibroblasts have contractile proteins like actin, which gives them the ability to contract. The actin filaments are in contact with a peripheral protein vinculin. Vinculin is attached to talon. Talon is attached to surface protein integrin. In turn, integrin is in contact with fibronectin, an extracellular protein which makes contact with collagen. This gives fibroblasts the ability to transfer its contraction onto the collagen fibers. Now coming to resorptive cells. They have numerous mitochondria and Golgi apparatus but fewer rough endoplasmic reticulum. Osteoclasts. Osteoclasts are the bone resorbing cells which develop from monocytes. In the periodontium, they are present on the surface of alveolar bone proper. They are large multinucleated cells located in small concavities called house-shaped lacunae. They have ruffled border which releases enzymes and hydrogen ions towards the bone for bone resorption. Cementoclasts. Cementoclasts are cementum resorbing cells which develop from monocytes. In the periodontium, they are present on the surface of cementum. Now, cementoblasts are not that commonly seen as, unlike bone, cementum is not remodeled continuously. But just like osteoclasts, they are also multinucleated giant cells but only smaller in size. Fibroblasts. Fibroblasts have a dual action of formation of collagen as well as their destruction. Fibroblasts carry out both extracellular collagen degradation and intracellular collagen degradation. In the periodontal ligament, if we take a look at collagen degradation extracellularly, fibroblasts have enzyme like collagenase, which when released outside degrades collagen. On the other hand, intracellularly, the fibroblasts have enzymes like stromalysin and matrix metalloproteinases, which on being released outside cleaves the triple helical structure of collagen fibrils and then phagocytose them into phagosomes. These phagosomes then fuse with lysosomes to form phagolysosomes, where enzymes like lysosomal cysteine proteinases degrade them. Epithelial rests of Malassez. These are dormant epithelial cells. In the periodontal ligament, they are present in clusters towards the cementum around 25 microns away from it. They are small cuboidal cells with scanty cytoplasm and a deeply stained nucleus and are surrounded by a basement membrane. If we look at their development, during the stages of tooth development, the Hertwig's epithelial root sheath initiates radicular dentine formation and then starts degenerating. The remnants left behind form the epithelial rests of Malassez. Defense cells. Cells like mast cells and macrophages are present in the periodontal ligament. Progenitor cells are undifferentiated mesenchymal cells. In the periodontal ligament, they are located mostly adjacent blood vessels. They are small cells with scanty cytoplasm and can differentiate into any of the progenitor cells when required.